I'm Yumika Kaneko from Tokyo University of Agriculture and Technology. I'm a master student at this university, and I'm interested in lipid metabolism in a diatom called Fistulifera solaris and biofuel production. I'd like to present about the psychological role and structural arrangement of the mitochondria of this marine diatom. In our study, we are focusing on the Fistulifera solaris, which is an oleaginous marine diatom that have high lipid content and high growth rate. We succeeded in 640 kiloliter scale outdoor cultivation of F. solaris, and its genome information and transformation techniques are available. So, F. solaris is a promising candidate for biojet fuel production by gene modification, and increasing the lipid productivity is required. In order to increase the lipid productivity, we are focusing on the attenuation of the oil degradation. To achieve the goal, elucidation of the oil degradation mechanism is required. Recently, enhanced association between oil bodies and mitochondria was found in animal cells, which are actively degrading oil. And the increased contact enabled efficient fatty acid transportation. Therefore, we thought mitochondria might directly contribute to the oil degradation also in F. solaris. For investigating the mitochondrial contribution to oil degradation, we conducted these experiments. First of all, oil degradation of F. solaris was induced by nutritional supplementation. After that, the culture medium was separated and oil bodies and mitochondria were stained every 24 hours. After staining of organelles, cells were observed using confocal laser scanning fluorescence microscopy. This is a 3D image of F. solaris. In this image, oil bodies, mitochondria, and chloroplasts are colored in blue, green, and red. As you can see, at first, tubular mitochondria scattered throughout the cell. On the other hand, when the oil degradation was induced by nutrient supplementation, the mitochondria was unevenly distributed at the both ends of the cell and tended to localize around the two oil bodies. Then we additionally conducted the image analysis to calculate the oil body volume and quantitatively evaluate the contact between the mitochondria and oil bodies in the cells. The contact ratio was calculated by dividing the contact area by the surface area of entire oil bodies. As a result, at 0 to 24 hours, when the noticeable oil degradation occurred, contact of both organelles increased. This means that mitochondria seems chase oil bodies and increase the contact with it, along with the oil degradation. Finally, we conducted transcriptome analysis of fatty acid degradation. During the oil degradation induced by the switching from nutrient poor to rich, the gene expression involved in the mitochondrial fatty acid degradation was upregulated. These results suggest that gene expression behavior is consistent with the appearance results which I showed you before. In conclusion, under inducing oil degradation, when the oil body was shrinking, F. solaris showed increasing contact between mitochondria and oil bodies. And we also confirmed the upregulation of the genes involved in mitochondrial fatty acid degradation. Therefore, mitochondria likely assemble the contacting phase with the oil bodies and directly contribute to the oil degradation. As a future prospect, we are going to identify the knockdown or knockout targets to decrease the contact between mitochondria and the oil body, leading the enhancement of biofuel production. In addition, we also proposed the importance of F. solaris as a model for the analysis of organelle dynamics due to its simple structure composed of one chloroplast, two oil bodies, and unevenly distributed mitochondria. Thank you.